Well, let's go ahead and get started. My name is Margaret Leinen. I'm the Director of Scripps Institution of Oceanography at the University of California, San Diego. And I also chair the Science Advisory Committee for the Ocean Biomolecular Observing Network, which is sponsoring this fifth spotlight on our Ocean Decade projects. And we're going to start off with a brief presentation that's an overview of OBON, since we're recording these for YouTube, and people may come across them who don't know about the network. So I'm going to share my screen and go ahead with the presentation. So the Ocean Biomolecular Observing Network is a UN-endorsed program of the UN Ocean Decade for Sustainable Development. And it's sponsored by the Partnership for Observation of the Global Ocean. And if you want to know more detail about the Ocean Biomolecular Observing Network, you can find our strategic plan at our website, and it outlines the strategy that we're going to use to observe biodiversity and marine life over the period of the decade. So our vision is to accelerate informed decision-making to restore the health of our ocean using the universal signatures of life on Earth, biomolecules. And our mission is to create a hub for biomolecular measurement of marine life. And the program envisions using all sorts of sampling devices, like the ones seen in this illustration, to obtain water samples from the ocean in which biomolecules from the organisms living in that water are located, analyzing those biomolecules to understand what's been in the water. So our objectives are first to build a coastal to open ocean multi-omics biodiversity observing system, and to do that through developing autonomous sampling and analysis capability and promoting methods and technique development, as well as sampling from more conventional devices, just sampling from ships, sampling from moorings, etc. We also want to enhance marine omics data systems and enhance ecosystem models by adding biomolecular components. We'll partner with marine conservation and management specialists, policymakers, and the private sector to address sustainable development using these tools. And then we want to of course, develop and transfer capacity through training programs combined with funded equipment programs supported by development aid agencies and philanthropy. Our strategy then is to inform decision-making by innovating, developing new technology for omics analysis, enhancing that through models like digital twins, data management, and equitable information use, and then develop our networks and resources through philanthropy, funded equipment, etc. So these three tracks will lead us to that ability to inform innovative solutions, ocean health, ocean management policy, all for a sustainable future. Our program has 20 UN Ocean Decade endorsed projects, and we're going to hear from a couple of those today, and I'll introduce those at the end of this brief presentation. This map shows our current sampling locations. Some of these, most of the ones you see are in coastal waters, and most of those are time series analysis. But we're starting to get data from cruises undertaken by our projects, and we expect that to be growing in the near future. We're organized through a program coordination office that is under the umbrella of the Partnership for Observing the Global Ocean. And this coordination office does planning, internal coordination of our projects, national committees, and so forth. A science advisory committee and an executive committee chaired by myself provide science direction and overall executive coordination. And later this year, we'll be starting a user advisory committee from users in all sectors that are interested in using ocean biomolecular observing. We have 4 working groups, one on technology development, one on capacity development, one on protocols, and this fall we're setting up a new working group on policy. Some of the workshops that we have had and are planning include an eDNA workshop at the Westpac meeting in April of this year that had 80 participants. We're in the planning phase for a workshop on fisheries management using eDNA that will be next spring at the Rome FAO offices. And one of our national groups, country groups in Colombia is developing a Latin America eDNA network and capacity development workshop for 2025, and that will be sponsored by Invamar in Colombia. So here are ways to contact us, a website that is obonocean.org. You can also contact our POGO lead, Sophie Siev, or myself, and you can follow us on at OBON Ocean on X, and also follow us on our website. With that, I'm going to stop sharing and start the program, which is looking at a couple of our projects that are observing systems. And first, we're going to hear from Karen Tait of the Plymouth Marine Lab, and she's going to tell us about AMT-omics, and that stands for Atlantic Meridional. No, I've gotten it wrong, Karen. I'm going to let you unpack the AMT-omics, and I'll turn things over to you to describe this new project that we're very excited about.
Okay, thank you, Margaret. I'll just start sharing. Okay, you can see my screen, the presentation?
Yes.
Great. So, yeah, my name is Karen Tait, and I'm a molecular biologist here at Plymouth Marine Laboratory. I'll be telling you about a freshly endorsed project called AMT-omics, which brings together the main producers of molecular samples that have been taken on board cruises of the Atlantic Meridional Transect. So I'll first tell you about the AMT program, all about that, and then I'll go and tell you a little bit more detail about what we'll be doing within AMT-omics. I'm the main contact for the AMT-omics aspect, but Andy Raes, who's also based here at PML, is the program lead for the AMT. So AMT, the program actually began back in 1995. So it's been going for quite some time now. And in that time, it's completed 30 research cruises with 4 more cruises planned in the next few years. So it started with a group of scientists taking the opportunity of a UK research vessel going from the UK down to either the Falkland Islands or the southern tip of South America every year to refuel the British Antarctic base. So it started with just taking samples as the ship was moving underway. But since then, it's developed into much more of a program with more time taken at stations to collect samples. During the history of AMT, we've estimated there have been over 250 scientists involved on board. They come from over 60 different research institutes, and there have been over 300 scientific papers published. A key ethos of the AMT programme has always been about supporting early career researchers. One of the things that the AMT programme particularly is proud of is that over 100 PhD students have either directly participated in a cruise or they've used the AMT data as part of their PhD studies. So these are just some of the faces that have been involved in AMT over the years. This Glenn Tarrant here, I think he probably has the record for most AMT participation. So over the years, it has been funded in various ways, but the key point is that funding has been secured just last year, and it will continue until 2030. With a full intention to secure more funding into the future. So the main objectives, well, a lot of samples are collected to look at the plankton ecology. Our main measurement is always flow cytometry, but we also, depending on the scientists on board, we've had people do detailed studies of zooplankton. We have people looking at pigments. We have a lot of biogeochemical measurements, oxygen, nutrients are always key. The main selling point of the AMT is this very long-term time series now, and the fact that it goes from UK waters all the way down the Atlantic to the southern tip of South America. Through the time span of the AMT, there's been a lot of great science that has been conducted. Some of it, for just a few highlights, have been used to ground truth satellites, satellite missions. There's a lot of work that goes into ecosystem model development using AMT data. And also, as I've also already said, there is a lot of effort in training the next generation of scientists. All the publications that have been linked to AMT, you can go onto the AMT website and see all of those there. They're very good at keeping that up to date. So although the personnel on board the ship changes every time, there are a series of measurements that are always taken. As I said, the flow cytometry is a key measurement. Optics, so suspended organic matter, primary production, detailed carbonate chemistry, nutrients, oxygens, pigments, and increasingly the nucleic acid samples that we take using a Sterevex is also becoming to be a key measurement. Again, on the AMT website, which is really very well maintained, you can find links to every single cruise report from the AMT, which details what measurement was taken, when it was taken, the time it was taken, the method that was used, the scientist who did it, so you can really trace back exactly, who did what and when, which is, a fantastic resource. I mean, this is some of the data. So this is Gavin Tilstone's primary production data comparing micro and picoplankton across one of the cruises. Glenn Tarrant's flow cytometry data, so you can see, the kind of detail that you can get across a vast geographic area. all of this data, with the key exception of the molecular data, where this is where AMT-omics, the idea was born really from that. All of the data currently is stored with the British Oceanographic Data Centre. And you can go to the BODC website and pull down any of the AMT data sets. They have told us, you know, that the downloads from of these data sets across the globe is just phenomenal. So we know that the value of the data that we have and just how much it's used by the scientific community across the globe, which is a shame when all those molecular samples and all the work that's been done with molecular samples isn't included in this. And so this is one of the key things that we want to address through AMT-omics. So working with the key partners who have collected molecular data over the years on the AMT programs, we want to remedy this. We want to make our data more accessible and available to the scientific community, which would be fantastic, particularly when you consider all the metadata that's collected at the same time as these samples. So, you know, what you can do with that scientific data is, you know, the possibilities are endless. We also, knowing the value of that long-term time series, but the fact that it's been collected not just with different molecular methods, but different sometimes morphological methods, sometimes molecular methods. We want to see if we can integrate data sets to create very long-term time series. And also key part of what we are trying to do is continuing with AMT's ethos of supporting early career researchers. So at PML, there's a small team that anyone coming to the OBON meeting in early November, you will meet the small team of molecular biologists and bioinformaticians based here at PML. But overseeing all of this as well is Andy Raes, who leads the AMT program. So I've already made a start of collating together all of the molecular samples that we have. So we're going to create an inventory, not just of the data that is available, but of the actual samples as well. So there's a lot of samples that are in freezers across the globe, which would be really, really nice to make these available for anyone who has funding to do something more with them in collaboration. So we'll make all this information available on the AMT website, and working within the excellent frameworks that have been put together within the OBON community already. We'll be creating a framework for how we manage our data going on in the future, with the aim of making it much more accessible, both in the raw formats and also in formats which are completely accessible to anyone, so non-molecular biologists. So to date, that data is from 11 cruises, 1,200 samples, likely more, and more cruises planned. Looking at how we might start to integrate data, those samples, they've been collected using lots of different methodologies, different sequencing techniques. Having seen what our next speaker, Eric, has managed to do with data collected from completely different sequencing technologies, I know that this kind of work is possible. So that's something that we'll be looking at. We'll also be doing a little bit of work to see how we might be able to integrate data collected through morphological means or from flow cytometry with molecular data sets. So one of the pressures that we have within the UK is to make our research cruises a little bit more carbon neutral. And that is pushing us down the route of automated sampling, which means that there is likely to be more reliance on molecular samples going into the future. But we really need to understand, if we want to create these long-term time series, how we can integrate it with the methods that we've used going back to 1995. So one of the key ways in which the AMT program has included early career researchers is through a partnership with POGO. I think on each cruise since, I think it's 2008, there has been a fully funded fellow from a country with a developing economy invited to work on board. We were very much hoping to have someone on board this current cruise. The next one's going out in November, who will be doing the molecular biology sampling and then coming back to PML to learn some more, a little bit about the molecular biology techniques. But we are having a few hiccups with the visa. So fingers crossed we can still make that happen. So that's the AMT-omics program, what we plan to do. And, you know, we will be very much using the opportunity of being within the network for guidance, and we're very open to share our own experiences with what we're doing with our data as well. Collaborations are very, very welcome, so please get in touch whether you're interested in participating in future cruises. We're very keen to explore training opportunities for younger scientists. I'm also interested in, if anyone wants to do anything with the data, you know, please do get in touch. And lastly, one thing that I think that I don't think we're alone in this, in that we could do with a little bit of more help in bridging the gap between our science and policymakers. So it's great to hear that that is an area that the OBON is going to be working on very soon. And I'll stop talking now.
Wonderful, Karen. That was a great description. And that transect of the entire Atlantic and long-term is just such a valuable resource. And we're really, really happy that you have taken on the job with your colleagues of really curating all of that data to make it fair, and also putting it in under the OBON umbrella. So we look forward to working with you. We're going to go ahead with the second presentation and then do questions at the end, since there may be some that relate to both of the programs. So our second UN-endorsed project that we're going to talk about today is the OceanOmics Monitoring and Protecting Ocean Life project of Minderoo Foundation, and Raes is going to describe this for us. Eric.
Thank you very much, Margaret. I'm sharing my screen, putting it in presentation mode. It's going a little bit slower on my end. There we go.
Perfect.
There we go. Hi everyone, my name is Raes. I work at the Minderoo Foundation. This is a philanthropic organization based in Perth, Western Australia, founded by Andrew Forrest and Nicola Forrest back in 2001. The vision of the foundation is a society that values all people and natural ecosystems, and our mission is to forge a fairer future. I would like to acknowledge first that I'm giving this presentation on Wajak Pujar country, and that the Noongar people are the traditional custodians of the land here. I would like to acknowledge their role as the first marine scientists and innovators. Also, a big shout out to Fiona and Luz for organizing the webinar series. So I'm a researcher in the OceanOmics team, where we use environmental eDNA or marine genomics as a non-invasive, rapid, and scalable tool to monitor marine wildlife with a focus on marine vertebrates. In our team, we kind of crossed pretty much the entire workflow of marine genomics, with colleagues focusing on the molecular innovation side, eDNA-based forensic approaches to detect, classify, and enrich species, populations, and individuals from seawater. Our fantastic computational biology and AI team are looking at new ways how we extract information from eDNA, including new AI tools and data visualizations. We have our ocean genome team, which are filling in the needed blanks in the reference genome libraries of all marine vertebrate families and threatened species. And then we also have our focus area scaling at sea, close to my heart, where we are funding and supporting the development of ship-based and, as Karen just mentioned, these autonomous platforms to enable this ecosystem scale deployment of eDNA. Across these 4 focus areas, we also have impact projects where we're really focusing on how the data and the eDNA and information that we get from it can be translated into application policy. And we're working closely with the Australian government through Parks Australia, Musea for the reference genomes, industry, and institutions. One key example, kind of our flagship, as an example where we're working across that entire workflow, is a partnership that we have with the Australian government, Parks Australia, where we're investigating and providing a proof of concept how eDNA can be deployed as a cost-effective way to monitor biodiversity in Australia's marine park, really large scales. Specific focus here, as Karen also already mentioned, is kind of tackling that translation to policy. And that's where we're sitting together with park managers and thinking about how we can help define how this eDNA monitoring data can be implemented into management, policy, and regulatory frameworks as well. How we are operationalizing kind of or providing this evidence-based science is through dedicated expeditions. This is a figure from 15 expeditions that we've completed to date. The colors are just different expeditions. The circles are samples taken in shallow waters, everything around 0 to 50 meters depth. And the pink squares that you see over here are samples from the deep sea, collected in collaboration with the Deep Sea Centre at the University of Western Australia, where they deploy these deep sea landers, which have baited cameras, baited traps, cameras, and miskin bottles, which close and then collect the eDNA and a variety of sensors, ADCPs, CTBs. The green polygons that you see on this figure here are the Commonwealth Marine Parks from Australia. And zoom in here a little bit in the different expeditions because Australia is a big country, and at the moment we have data from the tropics, Cocos, Christmas Island, all the way down to Tasmania, pretty much a distance of 6,000 kilometers. To put this in perspective, from the top of Norway to Portugal is 4,000 kilometers. So we're talking about vast distances here. Temporal data is usually important. Next year, we will have the 5th annual eDNA survey from the Abrolhos Islands. These are 3 islands, actually a group, it's an archipelago of 122 islands, but we surveyed the 3 big ones, sitting at this nice transition zone between tropicals and cold waters. These islands are heavily impacted by marine heat waves, specifically during La Nina. The last 2 years, we've also been working with the local indigenous community, the Yamaji Corporation, having traditional sea rangers on board on our expeditions and having that really nice two-way knowledge exchange. All the data that we collect from these expeditions is coming to our lab at the University of Western Australia, where we have dedicated short and long-term sequencing technologies, long-term more so for the tissue samples to fill in those gaps in the reference libraries. And from there, it's off to the bioinformatics, the crunching, and kind of that visualization part. This is a transect that we did from Perth in 2022 to Cocos Island. Nice latitudinal transect where we kind of asked the question whether we could pick up eDNA traces from pelagic fish and whether we could identify known latitudinal variation. This figure here on the bottom is kind of our sampling size from 1 Perth all the way to Cocos Keeling to the left. And at each site, we collect 5 replicates, 2 liters, filtered through 0.45 micron. The size of the bubble indicates how many times a fish family is present in a replicate. So if a fish family is present in all of our replicates, large bubble. If it was only present in 1 out of our 5 replicates, we don't even bother showing it on this map over here. You also see a little photo here under the map from our dedicated OceanOmics container. And what we kind of see is that we do see a nice latitudinal gradient from temperate species closer to Perth, and these more tropical species, such as flying fish, really start to pop up on the Tropic of Capricorn. We also see some cryptic deep sea fish pop up along the Transect. And of course, what we'd expect is kind of Myctophidae across the whole Transect. So it kind of aligns with the conceptual understanding from eDNA. But there is a lot more. eDNA data really has the power to contribute to global observations. The figure that we see here on the right-hand side is a compilation from observations from OBIS fish sightings, and we obviously see some gaps here in the Eastern Indian Ocean. Most of the observations are closer to shore, as you would expect. But when we combine the observations that we see in the sightings from OBIS, together with our eDNA occurrence data, we do see some promising results. So the light blue is data extracted from this polygon from OBIS, and the dark blue is our eDNA occurrence data. And so we recorded a sighting, so in the dark blue, for our eDNA as a unique eDNA detection that appears in at least one replicate. So if a certain fish family was present in all of our 5 replicates, we still acknowledge it in that site as just one sighting. And we do see already that these occurrence data are contributing to our observations and are able then to potentially fill in some of these gaps. One specific example that stands out here, these are 2 different mitochondrial fish markers, 12S and 16S, but are these telescope fish or Giganturidae. In OBIS, there were only 5 observations, but we recorded 55 eDNA occurrences, really showing that there is potential to integrate eDNA observations, absence, presence in this kind of global repositories. The power of eDNA to contribute to global observations. When we go on our expeditions to on country, Cocos Island, Christmas Island, the Abrolhos, we really make sure that we connect with the local community, the schools, the local rangers, fisheries, park managers, to like talk about ocean advocacy, ocean literacy. Here we engaged with 122 students. We extracted eDNA from bees. That was a little bit easier than bringing a dead fish to the classroom. But that, together with bringing the rangers out in the field, especially the fishery rangers on Cocos and Christmas Island, allows us to ask questions, what would you like to see from our eDNA data? And now we're starting to look into kind of that like policy engagement and visualizations. So the rangers, they really asked us, like, can you show us this Maori Raes? Because it's a critical and important species that they fish for. And on the left-hand side, you see a picture from Cocos Island, 2 surveys in 2022 and 2023. The crosses are sites where we collected DNA but didn't see it. And this is kind of the information we bring back to country in Christmas Island here specifically as well. People are really interested in like the wahoo and see like, also we actually fish on that side. Why don't you see it? And it's really these like simple questions that we want to dig into. But then we show actually more data and go like, well, we have 2 different genetic markers, mitochondrial 12 and then like 16S, and we do see differences in the detection in our replicates. And it's bringing people along for the ride and exploring molecular genetics as well, even if people are not trained into it. Some other data now at a much finer resolution is data from atolls in the northwest of Western Australia called the Rowley Shoals. Now we're talking about a fine scale resolution, 50 meters, where we collected DNA data on the slope of the reef and inside the lagoon in a multi-dimensional principal component analysis. The fish community really pulls nicely apart between these different habitats. Not something that we would expect, but again, it's like showcasing that we can track variation across hundreds of kilometers down to 50 meters with eDNA. We can use effect sizes then to pull apart the different family of the fishes between the lagoons and the slopes, and dig into this fine scale resolution and ask questions, where would we place boundaries for marine protected areas? And it's not only the lines, it's also the diversity within that is important. And so suddenly from a fish community diversity perspective, we can look at the amplicon sequence variants or haplotypes for one specific species. And that's what you see here on this figure. The color codings are the different atolls, Montebello, Imperius, Mermaid, and this kind of dendrogram shows the different ASVs. for this, or the haplotypes for this specific species. And what you might even pull out, here is this, all the red ones are all different samples, only occur at the Montebellos, hinting towards some kind of like endemic ASV haplotype for this little tiny silver site. The main crux here is that we can use eDNA not to define boundaries, fine scale resolution, but also highlight the diversity that we need to preserve within ecosystems. And that diversity at the haplotype level or amplicon sequence variant is really different between species, of course, but it is nice again to like visualize and get an understanding. Lastly, an expedition which I wanted to highlight is along the west coast of Western Australia. We did 2 expeditions here. The West Australian government would like to place a marine protected area. This is a stretch of 1,000 kilometers. And again here, eDNA surveys allows you in 10 days to stretch kind of like huge area and visualize the kind of hotspots or the presence, absence of certain fish families. Along that same transect, we also did a comparison between traditional nesting sampling and a semi-automated eDNA sampler from Ocean Diagnostics, a company based in Vancouver, which is able to filter water at depth with a variety of metadata, CTD integration, latitude, longitude, everything is logged. And so the symbols show the different methods and the colors, the different sites. You see already really nice like site fidelity around it, a little bit of overlap. These were sites really close to each other. Again, promising data that we can start to use automated methods, which then will increase our reliability, reproducibility, because it will all be done by the same machine, and also allow us to scale the collection of eDNA. So where are we going? We continue to work very closely and collaboratively with Parks Australia. These are one of our main stakeholders in translating that eDNA to stakeholders on the ground. The scale of eDNA is rapidly growing. So in the lab, we're automating, and in the field, automating as much as possible to get that reliability, that reproducibility of our data. And we're also starting to use AI to kind of like synthesize this big eDNA data. An example of the AI is our dashboard. Was online, we just took it down. We're doing some quality control and curations, but we'll be launching it soon again. It looks a bit like what you see over here. You can click on our stations and explore the data, the species with some photos. We won't be including IUCN species that are listed as critically endangered or vulnerable. If you would like those data, you can get in touch with us. And what we also have is this kind of like chat with our data. This is a large language model or a GPT where you can start to ask in human language questions. Are the sharks in the data? Can you list where a parrotfish was detected and then list where these species were found, for example? And you can just add a chat like a normal chat GPT, but it also gives you the thought process in a Python script, and then you get an answer. Yes, the fish was found here. And it's just another way so non-specialists are able to interrogate the data as well. We're also using AI to summarize our data for stakeholders. It will give you a list of how many sites were there, the years, and an AI-based summary, for example, in which certain ecosystem habitat the fish were found. That same large language model also incorporates external data sources, such as Aquamaps, a database for fish distribution probabilities, Fishbase, a database collecting which species are native to a certain country. The large language model also has knowledge of which species occur where. It includes the Australian faunal directory, and all of that information from these different databases is then allowing us to group our eDNA occurrence or sightings into 3 different groups. The first one is a group that is validated species, highly likely to be there, confirmed by all the different databases. The second group are eDNA occurrence data that are plausible, species which only occur in certain databases with a high identity or high probability from our blasts. And the last one are potential close relatives. And that also gives stakeholders a little bit of confidence. These species were certain here, these maybe not. All of our coding and pipelines are available on GitHub. I'm sure the presentation will be shared, so have a look at Minderoo Foundation OceanOmics Amplicon pipeline. It's version 1, and the large language model for the ChatGPT is also open source and freely available. 2 slides which I would like to highlight is kind of like how do we scale? And we're very close to signing a contract with CSIRO, Australia's National Science Agency, where Minderoo will be funding the R&D of an eDNA autosampler, which will be installed on the RV Investigator for 3 years. And Minderoo will be sequencing at least 2 amplicons, mitochondrial 12 and 16S for marine vertebrates. And so that really comes back to what Karen was saying, creating these longitudinal data sets that can be like all brought together in fairing care principles. We will also be hosting the second Marine Omics Technology and Instrumentation Workshop. It's a long name. This is a spin-off that was organized by the Technology in Development Working Group from OBON, organized 2 years ago at Mbari, and we'll be organizing that next year. Leveraging current infrastructure and incentivizing cost-effective eDNA surveys on existing platform will really be essential to ensure that we ensure the widespread adoption. A last example that I would like to highlight is that Minderoo is supporting an eDNA hub here on Christmas Island, pretty remote, and we will be operationalizing an end-to-end workflow. We will be giving our eDNA container on loan. We will be giving the park rangers the Ocean Diagnostics eDNA sampler, and we'll be kitting them out with an extraction kits and hopefully an iSeq as well, along with bioinformatics support on island. So with that, I would like to thank a whole lot of people, researchers, collaborators, and of course, Minderoo chairs, Dr. Andrew Forrest and Nicola Forrest. Thank you very much.
Oh, thank you so much, Eric. That was terrific. Fiona Beckman has dropped the links for AMT-omics and for OceanOmics into the chat, so you can have a quick path to the websites. And with that, I will open things for questions. Frank.
Hello, everybody. Thanks for the really wonderful presentations. Really exciting to see these time series and broad scales of sampling. I don't know if Eric, if you mentioned it, but also Karen, I was wondering to what extent is your intent is to follow the GBIF and OBIS protocols for managing the data and sharing the data with the Darwin Core data formats?
So I'm also involved in the OBON. So that one's based in the Western Channel Observatory. And I have gone through the process with our data manager here at PML of how to go about getting molecular eDNA data into OBIS format. So we've done it once. And once we've got our AMT data into a good format, then we know the process now, so we will be doing that.
Great.
Same for us. Our computational biology team are already closely speaking with OBIS, Darwin Core, GBIF, to kind of like have everything uploaded. They're trialing some templates and kind of, yeah, everything is, as we speak, ongoing, and it will be uploaded, fair and care all the way, and absolutely to OBIS, GBIF, Darwin Core.
Great, that's wonderful. Thank you.
Just to add to that, if I may, Frank, we're having this OBON annual meeting. Sorry, this is Sophie from Pogo. I'll try and show my face. I
recognize you.
Yes, so we're having our annual OBON meeting in a couple of weeks in Plymouth, and tagged on to the end of that, there will be a half-day workshop run by Sarah Swoman from OBIS, trying to help OBON projects to format their data to get it into OBIS. So it's great to hear that Karen's already been through the process, and yeah, so she
that is fantastic. I'm really very happy to hear. Very happy to hear that. And your programs are helping advance that interoperability that is so critical to get. Eric, you use the OBIS database to help validate your data, and one of the problems that we find with the OBIS records is that they're not growing as fast as they should if we want to monitor global biodiversity or even regional or national. And so the kind of data that both of your programs provide and all of the OBON programs provide is really very critical.
Sophie, we should look into whether we could do a video of that training session for our capacity development effort.
Yes, and maybe OBIS already have, I know they've been working on various, I think they're developing a training course for OTGA, is that right? Ocean Teacher Global Academy.
They do, they have several already, but I don't know if it's specific to the DNA, to moving a DNA, so, but I can find out.
But yes, we'll definitely record anything we can and try to make that available.
Great.
I do have a question as well, if there are no others, to Eric, kind of picking up on a comment that Karen made about the Australian. I think there was a publication recently where you were looking at data sets from data that were obtained using different methodologies or different sequences and things like that. Could you say a bit more about that and what you did and what the outcomes were?
Yeah, this all started during the pandemic. Karen and I had never met, and I reached out to her, and it was amazing. First, would like to say that like scientists are extremely open to share data if you ask nicely. But so we're looking into time series data. Australia has 7 national reference stations. Jody van de Kamp is championing those, where eDNA data has been taken for the last decade every month. During the pandemic, I was working remotely for the lab at Julie Laroche at Dalhousie University in Halifax, and they had a time series going on for 3 years, weekly sampling. And so I started to reach out. I used to work at the Alfred Wegener Institute, and then these people got involved, time series up in the Fram Strait. And then it started to grow with the time series run up by Feb Gasol in the Mediterranean. Spots came on board as well. And so everybody has different amplicon. This was just purely for bacteria, but even then, different hyper-valuable regions, different volumes, different extraction chemistry, different preservations. Everything was kind of like different. And even the time series at PLM was analyzed with 454 sequencing, pyrosequencing. It was even really hard to get to the raw data. So we're talking with like time series with very like different. But when we look at the seasonality of the community of the bacteria, we saw some really strong signals that were across all the different locations, from the Fram Strait in the Arctic to California to Tasmania to England. And so what came out was that yes, kind of standards around metadata and the availability of the data, it's usually important, but we are still able to extract meaningful seasonal trends, even though we have all different methodologies. And I think that's why the AMT-omics is also so special, because it's it's data across a vast spatial scale, but also across time. Although the methods might be different, we will still be able to pick out patterns.
Thanks, Eric. And Karen dropped the link to the paper into the chat. Thanks for that.
Yeah, I think now that I just because I just had to think a little bit. One thing which we couldn't do was actually compare absolute diversities, because everybody was working with different primers. And so when we start to align primers and hypervariable regions and like have more kind of like standards, then we will be able to actually quantify that change. Now we were just able to kind of show there is a change.
Karen, it looked like for the Transect data that you go down to about 300 meters. Is that correct?
Yeah. So the measurements they tend to go down to the deep chlorophyll maximum, and they don't go any further than that. So it is just the top 100, maybe 200 meters that we have. Most of the PML data that we have are surface samples as well. So we have very few data from further down. Jed Furman, he tends to do much more depth resolved sampling.
And is the cruise usually at the same time each year?
It does tend to be towards the end of the year, so it is within the winter months. Yeah, this particular one, they're going to be still on board all through Christmas. I think they go into Argentina just in time for New Year's Eve. So it's a tough gig.
Frank?
Yeah, Karen, you mentioned that different people that go on the cruises use slightly different protocols. And I was wondering, so we have the same issue everywhere, of course, with eDNA. It's kind of a Wild West still. So maybe two questions. One is, how do you plan to compare or do a round robin, and so your time series are comparable across, so your samples are comparable across time? And this is a broader question for OBON, since we have such a still expanding universe of methods and groups of people using different methods, how is there even a possibility of having a common denominator in DNA methods so that at least a minimum of data is interoperable or comparable?
How we go about integrating that data, yeah, we have no idea really yet. I think that, until we sit down and actually see what it looks like, it's hard to know the approach that we will take. We're lucky, I guess, in that I think probably 80% of the meta barcoding has all been done with the same method of sample collection, the same sequencing, done at the same time. So, that's giving us a good starting point. But to start bringing in all the other samples that have been collected by other researchers, yeah, sometimes, so we go straight onto a SteriVex. We don't do any pre-filtering at all. But other groups like to fractionate. So they use the approach that the Tara Oceans approach. So it's, yeah. We'll be sharing as we go through this process, our thoughts and what we'll be doing with all of it.
So to ask Karen, the Germans from the Alfred Wegener Institute, they also have a lot of transects going from Bremerhaven down to Antarctica.
Yeah.
Yes, they will be keen to, maybe they have their own project.
Yeah, so interestingly, when this is the annoying thing when people are uploading sample data to NCBI or EBI, whatever they choose to use. Sometimes they're not even including the name of the cruise. That it's really difficult to find that information. But I had more success finding those German cruises than I did with finding the UK cruises. Yeah, so I'm aware of the data, and it's actually the metadata that goes with it is in a better state than what we have, unfortunately, for us.
Exciting to have like if you're able to locate the bioarchive samples to see what extra amplicons or metagenomes could be funded or like sequenced, in addition to all the other metadata that you mentioned that has been collected, it will give you a really nice picture.
Yeah, yeah. Yeah, and at least we know that that's in a very good standard of you know, it's been collated very, very well.
Yeah, we could maybe reach out to them from OBON and ask them if they'd like to collaborate,
Yeah.
become an OBON project as well.
Yeah.
If you know specifically who's involved, it might be different people on different cruises, but I don't know if there's one person who kind of oversees all the biomolecular work on those transects. We could try and find out.
Yeah.
And just going back to Frank's point, that it has come up a few times in OBON discussions, the idea of doing some kind of intercomparison exercise round robin type thing. So that's definitely something that's been discussed. Oh, Julie's just
Well,
posted.
if you think about that and making progress on that, we would like to work with you and support that effort. Specifically, I'm talking about the Marine Biodiversity Observation Network has several projects. Actually, I think all of the projects have some eDNA component, and we have this situation, where people need to be trained to mobilize the data, but also we have different groups using different methods, and so we've talked about a round robin type of effort, but we haven't done it. So it'd be nice to do it on a more broad basis with you.
With
Maybe
a ring
that's,
test. Sorry, Margaret, you go.
I was going to say, maybe that's a topic we could put on the agenda for the annual meeting. Just not as a big, lengthy discussion, but sort of get a consensus about it, and then put together a little ad hoc group that could work with those that are willing and design something.
Yeah, I know the conversation is also happening in the Glomicon group, and I don't know if they've advanced any. So there's several groups talking about the same thing, and I wonder how we coordinate ourselves, and maybe you are in the right position to do that.
Yeah, I think that's a great point. And if we got something started that was but that the diversity of OBON projects said, yeah, this makes sense, we might be able to push that into the other networks.
To some extent, and part of our own fault is, we have all these MBON projects that are collecting eDNA, and we haven't gone through the routine of writing an Ocean Decade proposal to become affiliated with OBON, but we are brothers and sisters in the same family. So wonder how we can, and I don't know to what extent your emails go to all the different projects that are doing this, and whether we have to go through the rigmarole of more decade proposals to become affiliated.
Well, we've been encouraging people that have other kinds of projects to be contributions, and that at least tags them so that we're aware and, get them on our email list and so forth without the hassle of writing a project proposal.
Yeah. Well, maybe I can work with you or Sophie to do that, and so we can have a group of projects that under the MBON umbrella, and that we work with to at least receive your notices.
That would be great.
Thank you.
And it does seem like it's a lot of the same people involved in all of
Yeah,
these
that's
different
true.
groups. Katie Pitts, I think Pierluigi is involved in Glomicon, and then there are quite a few people involved in MBON and OBON.
Yeah, I
So-
don't know why we do that to ourselves, but that's exactly right.
And the CSIRO ring test, has that been has there been any publications on that, or is it still underway?
The data have been analyzed. There are no publications yet. So what came out, I think 5 different laboratories in Australia, all sites from Tasmania, 3 or 4 sites, and it came out at the community level. All the sites separated really nicely in a PCA plot. But there is a lot to be able to dig into. So an interspecies variation, and so it's not as clear cut, I think. But I don't have these results. I can ask them and see if I can bring something to the meeting. You know.
That would be great, Eric. Well, if there aren't any more questions, I want to, Frank, Fiona dropped a place on the OBON website where you can sign up to receive the newsletter and updates.
Excellent. I'll share that.
Great.
Thank you.
So Karen
Bye-bye.
and Eric, thank
I
you.
need to jump off.
Okay.
All right.
Karen and Eric, thank you so much. Those were great presentations, and I think they really gave us a taste of what you're up to. And I hope that we can really promote the videos for all of these project summaries, because they're terrific to bring us up to date with what's going on in the projects. And we look forward to seeing you in a couple of weeks in Plymouth. And
Very
Eric,
much.
you're here too.
Yeah, thank you.
Yeah, thanks very much.
Bye.
both of
Thank you.
Thank you so much, everyone. Bye.
Bye.